In the lab this week, we studied the cell cycle, and the cell cycle includes interphase that cells go through, and also mitosis. But the purpose of the cell cycle and the purpose of mitosis is to enable cells to reproduce asexually, that is, without eggs or without sperm. So cells do mitosis in order to grow, or rather allow the organism to grow, and also to replace cells that are dead, or injured, or aging. Okay, so for this lab, I'm going to expect you to be able to recognize all the phases of mitosis in onion cells, that is allium cells, and also in whitefish. So it's important that you can recognize them all. And there are some quizzes on Blackboard that can help you study them. So let's start. At the very beginning of the cell cycle, the first phase is interphase. In interphase, the cell is preparing to divide. So basically, it's replicating the entire contents of the cell. And interphase is pretty distinctive. Most of the cells you've seen in the microscope so far have been in interphase. So I'm going to be using an allium slide in order to demonstrate the different phases of the cell cycle. So let's look at our video monitor. So this cell, for example, is an interphase. When you're looking at interphase, make sure the cell has a distinct nucleus with a nuclear envelope. So you can see there's a nucleus here, and that nucleus has got a distinct membrane around it. That's the nuclear envelope forming the nucleus. Also, if you look in here, you can see a darker spot. That darker spot is the nucleolus. Remember, the nucleolus makes ribosomes. So if a cell is in interphase, there's not going to be any visible chromosomes. The chromosomes will just be, or rather the nucleus will be dark in color, and you may be able to see the nucleolus. You'll see a definite nuclear envelope. So that is interphase. Okay, and during interphase, all the contents of the cell will replicate, including your chromosomes. So your chromosomes will go from being single-stranded to being double-stranded. However, you will not be able to see individual chromosomes. Once interphase is complete, the cell is going to be ready to start division. So the first phase of mitosis is prophase. In prophase, what happens is that nuclear envelope is going to disappear, and your chromosomes are going to condense and become visible for the first time. When you see them, they're going to look like little threads or little worms. So let's see if we can find some prophase. Okay, good. This cell is in prophase. If you look, you can, the nucleus is basically disappearing. The nuclear envelope is gone, and inside where that nucleus used to be, you could see individual thread-like or worm-like chromosomes. So if you see a bunch of individual thread-like chromosomes, and they're basically in a clump in the middle of the cell where the nucleus used to be, there's no nuclear envelope anymore, then that is prophase. After prophase comes anaphase. Anaphase is pretty easy to recognize. During anaphase, all your double-stranded chromosomes will line up in the middle of the cell. They're getting ready to separate and pull apart. Let's see if we can find some anaphase, or rather metaphase. Okay. This cell is in metaphase. You can see your dark thread-like chromosomes are in a line in the middle of the cell. Okay, they're no longer in that clump. They've actually spread out to be a line. You can clearly see some of the arms of the chromosomes. So after metaphase is complete, the cell is ready to undergo anaphase. And anaphase is the shortest phase of mitosis. During anaphase, those double-stranded chromosomes separate and pull apart toward opposite sides of the cell. So let's see if we can find some anaphase. Okay, right here is a good example of anaphase. You can see a clump of chromosomes here, a clump of chromosomes here, and they're being pulled apart away from each other. So that's a very simple stage. It's very short. You look for two groups of thread-like chromosomes being pulled apart. Now the final phase, telophase, is sometimes hard to see. In telophase, you see two groups of chromosomes. They're being pulled apart, and the cells are finally starting to divide. In a plant cell like the allium, you're going to have a cell plate grow up between the two cells. In an animal cell like the whitefish cell, you're going to see the cells pinch inward with cleavage furrow and finally pull apart. So let's see if we can find a cell with a cell plate. here is a good example of telophase. You can see a group of chromosomes here, a group of chromosomes here, and you can faintly see a cell plate starting to grow up. So that cell plate is a cell wall. It's going to grow up and divide those two cells, or rather that cell into two separate cells when it's finally finished growing. And so at the end of telophase, you're going to have two new daughter cells, and each daughter cell is going to reform and have its own nucleus again. The chromosomes are going to uncoil and no longer be visible. So for this slide, I want you to be able to recognize all phases of mitosis for both whitefish and for allium. Okay, our next activity, we'll walk over to the chalkboard and talk about what we did. Okay, our next activity was an attempt to estimate how long each phase of mitosis lasts. Basically, the idea is that on our slide, our cells have been killed, and so they're all frozen doing what they were doing at the moment they were killed. And if a particular stage lasts a long time, at any given time, there's going to be more cells doing it. If the stage is very short, at any given time, there's only going to be a few cells doing it. So we looked at 20 cells on the allium. We decided how many of those 20 were in each particular stage, and we got a fraction from it. So this is our group data for one of our classes. You can see this is for one group, another group, and so on. Here's our class totals. So out of 79 cells, 24 were in interphase, 20 were in prophase, 16 were in metaphase, 9 were in anaphase, and 10 were in telophase. So we just turned this into fractions, like 24 out of 80, 21 out of 80, 16 out of 80, and so on. And we multiplied our fractions by 16 hours, because 16 hours is the total length of time that mitosis takes place in allium. So if you multiply the fraction of cells in any one particular phase by the total length of time the entire cell cycle lasts, you'll get an estimate of how long this phase lasts. And so we found that an estimate of about 4.8 hours in interphase, 4.2 in prophase, 3.2 hours in metaphase, 1.8 hours in anaphase, 2 hours in telophase. So it sums up to be 16 hours like it should. And this is roughly correct. The longest phase of the cell cycle is interphase, and the shortest is anaphase, at least for allium. Okay, so for your final exam with your quizzes, make sure you can do simple calculations just by multiplying the fraction of the cells in one particular phase by the total length of time it takes. That is 16 hours for allium. Okay, let's walk on over and see our last experiment. For our last experiment, what we did was we made our own slide. We were trying to make a slide of mitosis in narcissus flowers. So this is a narcissus bulb, and we clipped off the end of the root. We soaked it in hydrochloric acid to soften up the cell walls. And we stained it with acetocarmine dye, which will stain the nuclei and the chromosomes red. Put the cover slip on the slide, and we just squished it with our thumb to make it flat and spread out the cells a little bit so you can see them more easily. Now this slide, unfortunately, usually does not work terribly well. Usually the only phase you can see is interphase. So very rarely you'll be able to see maybe metaphase or anaphase or something like that. So let's walk over back to the video monitor, and we'll see an example of some of the suits made. Okay, so this one came out as well as most of them did. We can't see really anything other than interphase. You can see some of the nuclei are visible for a view of the cells. And you can see cell walls and so on. Very rarely we'll get lucky and be able to see something other than interface. Okay, so that's the lab today. Make sure you can recognize all phases of mitosis for allium and also for whitefish. There's some study material on Blackboard that can help you do that, help you identify pictures, and so on. Also make sure you can estimate how long each phase of a particular stage of mitosis lasts. If you can count how many cells are